my screen. Okay, can you see the slides? Today we are going, so we saw all the genome structure, the chromatin structure. We saw a lot of things, especially on chromatin in relation to transcriptional activity, because if the chromatin is open, it can be transcribed. Otherwise, it's not working. So now we're going to narrow the analysis and just focusing on transcriptional activity and transcriptional regulation. So for this lecture, so maybe you will be a little bit surprised. So today we are going to go for a first part, which is mainly theoretical. So we'll go for, I will explain you all the types of RNA polymerase, the way the RNA are transcribed, and different modification. At the end of the lecture, we're going to start to see some of the method to identify gene elements in gene that are implicated in gene regulation and how we can analyze the transcription factor. And on Monday, we'll do an exercise. So it's a lecture, but we will take example from an exercise. Okay, so what I want to talk to you, sorry, there is regulation. Did you receive the PDF file from Miss Hang this morning?
Yes, we saw the email a couple of minutes ago.
Okay, perfect, because I sent it this morning quite early. Okay, so for transcript, wait, I put this full screen. Sorry, I've got plenty of windows.
Excuse me?
Yes.
Yes, for the previous lecture, when you give an example about the Drosophila flies, I tried to look for the exact paper that you used for the figures and the example, but I found all the papers from that researcher, but not that particular one in 1997. So if you have a copy,
which one is it? Which figure?
Is the figure that how can I show you?
No, I know. Okay, one second. So it was yesterday. So it was, I suppose, the last one where there was the red eyes. This one.
Yes, that one around like page 38, 40 something.
This reference at the bottom.
Yes, Walrath.
Walrath in current opinion in genetics and development. You didn't find this one?
Yeah, I tried to look up the author and the year, but I couldn't find any paper from him in that particular year. Just for other years and other papers from that researcher. So if you have a copy,
yes,
maybe.
I'm surprised. I note it down. I write it down. So what? Okay, and I'll tell you on Monday if I found the. I've got it, but I.
Yes. So, okay, that's okay.
Okay. It's noted. All right. But just to be clear about this figure plus this one, there are.
Wait, I can't see the slide. I saw the slide for lecture 3, but I'm not seeing the one for lecture 2.
You don't see. Okay. So the first. Okay. No, sorry, because it's complicated with Zoom because when you open two different. Hold on 1 second. I wasn't in Zoom. I need to reshare. So I need to stop sharing and reshare.
Can you allow other students?
Yeah. No, because I've got so many things that is coming in the screen, so I meet everybody. Okay. Is it this? Do you see the flies with the red eyes?
Yes.
So it's this figure plus the next one.
Yes.
This one. So just to point, this figure is not inside the paper. Okay, it's coming from the result of this paper, and this paper, Current Opinion in Genetic and Development, this is review papers. So it's a synthesis of plenty of different research papers, and all these pictures, they were taken from the references inside that review. So this is maybe why you didn't find it. But I will get you this paper. Is this one that you want?
Yes, I just want to read some more like text about the thing that is on this slide particularly.
Okay, so you want me to get you this reference?
I think so, yes.
Okay, so I'll get you that on Monday. Okay? Yes, tell me.
Excuse me, can I have one question for this slide too?
Yes, no, just 1 second. It's quite difficult to do the lecture in Zoom. Usually when I do the lecture in USTH, we can take a lot of time giving question, answering and discussing. So do not hesitate to ask me questions. So what would you like to ask?
Okay, so in this slide, there are suppressor and enhancer, but normally I understand the suppressor going to suppress the expression of the gene, right? So in this particularly, suppressor is for suppressing the expression of the heterochromatin or what? Exactly.
This is not, they are not suppressor of the phenotype of the red eyes. They are suppressor in the case B here. The gene that has been mutated, they are suppressor of heterochromatin expansion. And this one, they are enhancer of chromatin expansions.
Okay, so the mutation in figure B is have the effect of suppress the heterochromatin, and yes, the figure C is to enhance the expression of the chromatin, heterochromatin.
Exactly. Because heterochromatin is symbolized by this white box. So in this genetic context, the heterochromatin is increasing. Okay. So we have to take care because it's very precise. When we talk about this phenotype, for example, the one in C, you see that heterochromatin is expanding, but it's expanding in the context of this type of mutant. Okay? Do you follow me? So it means,
yes.
So it means that the gene that has been affected by the mutation is not an enhancer, is a suppressor. Do you understand?
Can you?
Okay, I will repeat because it's quite tricky with the words. So because it's important that you understand, we can go a little bit faster for the rest of the lecture. So the initial phenotype is this one. Okay. So technically we've got partial expression, okay, because the wild type gene is just at the borderline between heterochromatin and euchromatin, okay. So this is the initial state. You generate mutant from this genetic background, and you have two types of behavior: either a phenotype in which you can see that they suppress the phenotype, okay, because you revert to the wild type, okay, and another type of phenotype in which you enhance the phenotype. You've got almost completely white eyes. So what is written and what you have to understand when I say suppressor, it's the suppressor of the initial phenotype. And this is an enhancer of the initial phenotype. Is it okay for that? Therefore, if we take the example of the suppressor of the phenotype, the phenotype of this construct is partially red. You suppress this phenotype because now it's completely red, and the suppression of this phenotype is due to a reduced expansion of heterochromatin. Okay? So it means that the gene in this genetic background that has been affected by the mutation, and the mutation gives this phenotype, is an enzyme that is responsible for expansion of chromatin. Because when you knock it down, you reduce chromatin. Is it okay?
Yes, yes.
You have to take the things one by one. The starting point is this artificial phenotype. From that, you generate mutants, and you have got, when it's written suppressor, it's suppressor of the initial phenotype that was already artificial, because the wild type is red. Do you get it?
Yes, I get it.
Okay, so it's just a question of words. Suppressor of the phenotype and enhancer of the phenotype based on the original one. Okay? So we start the transcription. Wait, it is this one. Okay. Partition. So now you can, wait, because I've got so many windows, I need to close everything. Okay. So now you see the slide with transcription regulation written. Okay. It's okay. So what we are going to see today, the first part, I think it's important. You will say to me, okay, but we never use this and so on and so on. I understand, but in terms of just fundamental molecular biology, I think at the master one level, it's important that at least you have some information about the different RNA polymerase. Because you will see after we will only talk about RNA polymerase 2. Because RNA polymerase 2, you know it very well, is the only polymerase that is implicated in the transcription of messenger RNA, and then that are going to code for proteins. So most of the time, let's say 99%, all the paper that you are going to read, all the gene expression analysis that you are going to find, they are talking about RNA polymerase 2 transcriptional regulation. But you know, so this is, you know that for you from your graduation. So this is a very small table just to remind you one thing. This is what are the 3 RNA polymerase. So take care about one thing first. There are 3 RNA polymerase DNA dependent. Okay. So can you explain me what is an RNA polymerase DNA dependent? I just want that it is clear from the beginning, and after we can go on. Can you imagine a definition of what is an RNA polymerase DNA dependent? And the next question would be, do you know some RNA polymerase that are not DNA dependent? If you don't know, it's not a problem. Just tell me if you have any.
I don't know.
Okay. No, it's not a problem. So there is one very important thing. It's the more data we have, it's on plants. So we have 3 in eukaryote. We've got 3 RNA polymerase that are DNA dependent. RNA polymerase that is DNA dependent. You will say, okay, it's obvious. It's a complex of enzyme, because you will see there is plenty of partners inside that is able to synthesize RNA reading from DNA. So it's really trivial, okay? But there are, and we will see that on Tuesday, there are some RNA polymerase that are RNA dependent, which means this is some complex of enzymes that are able to synthesize RNA, but not in reading DNA, in reading RNA. Okay, and this is implicated mainly in all what we call post-transcriptional gene silencing. So we have to really separate these two big. There is no relation between these two classes of enzymatic complexes. What we are going to see today, this is RNA polymerase that are able to transcribe RNA from DNA, from the genomic DNA. Okay, so the common feature in all the eukaryotic cells is that there is 3 RNA polymerase. So this, you know that for a long time. We've got RNA pol1, pol2, and pol3. I just put in this chart so it can be a kind of reference for you the localization of these 3 complex of enzyme in the cell, what type of RNA, because you know that there are plenty type of RNA that these enzymes are able to synthesize. And there is just the last column is just a small example because it was very used to characterize the different activities of RNA polymerase. There is a toxin which is called alpha-amanitin. It's coming from a mushroom, which is a deadly mushroom, which is called Amanita phalloides. That contains this molecule, alpha-amanitin. And this is the target of this drug is RNA polymerase in general. But you see that they have different sensitivity to this drug. For example, pol1 is not completely affected by this drug, and there are different doses that can affect the sensitivity of pol2 and pol3. So it's just to show you that using drugs is very, very common in this type of research, because you can, when you use this alpha-amanitin at different concentration, you can separate the transcriptional activity between pol2 and pol3 and so on. So it's something that have been very frequently used. So we're going to talk today about these 3 polymerases. So keep in mind that we will spend something like 1 hour on pol1 and pol3. And all the rest of the lecture and the coming lecture next week will be only in RNA pol2 and the transcription of genes that are going to be translated into proteins. So I wanted for this BP10 teaching unit that we spend at least half an hour, something like that, just to remind you how RNA pol1 works, and the same for pol3, and what type of genes they are reading and what type of RNA they are producing, because I think it's important, because you will see. So we will go today. We will check for. I will show you how RNA pol1 works, pol3 works, and then we will spend quite a lot of time starting the analysis of RNA pol2 transcriptional activity. So. One general comment about transcription. This chart is very interesting to me. So you have a color code, yellow, red, and blue, which is related to which RNA pol we are talking about. On the left, you've got, sorry, it's in French, just the top, but I think it's completely understandable. So, on the left, you've got the transcriptional activity of the 3 polymerases. So you see it's written in %. So what they did to produce this kind of data, they quantified all the RNA that is produced by pol1, all the RNA molecules produced by pol2, and the RNA molecule produced by pol3. And you see clearly that inside the cell, you've got more than 60% of RNA present inside the cell that are coming from only the transcription from pol1. Pol3, it's only 40%, and pol2, which is very interesting for us, represent only 1/4 of all the RNA present in the cells. And pol2, you know that it's transcribing the messenger RNA. So think about that. All the RNA that is then translated into protein to give function or to manage enzymatic activities and so on, they represent only 25% of the total RNA content inside the cell. And then if you look in the genome, okay, so on the right, the chart on the right, so it's a little bit tricky to understand. So what they did, they took the genomic sequences and they tried to sort inside these genomic sequences what is among all the part of the genome that is transcribed. Okay, so we are not talking about part of the genome where there is no transcription at all. Okay, we are considering only the part of the genome that can be transcribed by one of the 3 RNA polymerase. So those data here, they can be, the unit can be megabase because it represents a size of the genome. So here what you see is that the part of the genome that is read and transcribed by RNA Pol1 is only 10%. But this 10% of the sequence transcribed by Pol1 represent at the end 60% of the RNA content. Okay, you understand how to read and to compare these two charts? Okay. If we take the case of the portion of the part of the genome that is transcribed by Pol2, it's almost 90% of what is transcribed in the genome correspond to genes transcribed by Pol2. But all these genes, once expressed, represent only 1/4 of the RNA content. Okay? So from that, what can you say as a kind of summary? The strongest transcriptional activity in terms of quantity of RNA produced in a cell is coming from Pol1, but the matrix, the DNA that is read by Pol1 represent only a small portion of what is read by all the 3 RNA polymerase. Okay, so when we look back the chart, Pol1, do you see for what it is coding for? It's pre-RNA for the 28, 18, and 5.8S RNA. Okay, so this RNA needs to be expressed at a very, very high quantity because they are part of the ribosomes. And the ribosomes need to be present in huge amount in order to be able to do all the translation of the proteins. So the quantity of the transcript coming from Pol1, this 60% represent only the RNA that are present in all the ribosomes inside the cells. So they won't be translated, but this is the majority of RNA. Well, one of the conclusions is that almost 1/3 of the RNA inside the cell is for ribosome biosynthesis. Okay, is it okay for this part? So you see the disequilibrium between the size of the sequence read by the polymerase and the quantity of RNA that is produced by the 3 polymerase. So what I wanted to show you, it will be very quick. So just one point before we start that, I want to ask you to remember all the data that we are going to see very quickly on Pol1 and Pol3. Okay, it's not really, but I think that you need to have that we can have this discussion, and anyway you can see you can have that for your own records. So I'm just going to talk to you a little bit about the structure and how is working Pol1, RNA Pol1, because it's quite peculiar. So this RNA polymerase only transcribe the rDNA, which is the DNA that is going to be able to be transcribed into ribosomal RNA. So what's happening in mammals, this is the best documented data in mammals for that. The structure of this little portion of the genome, because remember, it's only 10% of the genome that is recognized by polymerase 1. 10% of the genome, of the transcribed genome, okay, that is transcribed by Pol1. Actually, it's composed by big pieces of DNA that are around 50 kb, and that they are repeated in tandem, repeated. All these bits of DNA, where do you find them in the nuclei? Do you have an idea? Remember what we said. The Pol1 is in the nucleolus, the enzymes, okay? So do you have an idea in terms of genomic organization, these 50 kb repeats that contain this kind of sequence that are transcribed by Pol1 in the nuclei, where is it located?
Also in the nucleolus.
Exactly, because what do you know about nucleolus in the nucleus? What do you know about its function?
I remember vaguely that it is related to RNAs.
Yeah. So
tRNA and rRNA.
It's mainly rRNA. tRNA, they can be also in the nucleus. So what we see, because you have to think in terms of history, when the researcher a long time ago, they started to do microscopic experiment and they just look at the nuclei, the nucleus, you have the nucleolus, it's more dark than the rest of the nucleus. And this is, according to you, why it is darker when you look in microscopy, the nucleus. Do you have an idea about that? Because everything is in relation from what we see now. So inside the nucleus, so we've got heterochromatin, euchromatin, and you've got this big spot. They can be one or two. It depends on what is the section for the microscopic experiment. You've got one spot, which is the nucleolus, and it is always darker than the rest of the nucleus. Why do you think this part of the nucleus is darker when we look at that under microscope? Do you have an idea?
Yes, because it's a site to produce ribosomes, so there are accumulated a lot of protein in ribosome.
Exactly.
So is it darker?
It's not because of the protein, but you are completely right. What is happening is inside the nucleus, you've got all the chromosome, and during the interphase, all the chromosome, they are expanded, okay, so the transcription can be active. What's happening is that all the genes, which are these 50 kb repeats that contain the DNA sequence to produce the ribosomal RNA, they are all located in the same place inside the nucleus. And because this, this is a small portion of the genome that is transcribed, but it is the portion that is transcribed the most. So what's happening is at that place in the nucleus, where there is the 50 kb repeats, you've got so much transcription from Pol1 that the concentration of rRNA is extremely high, so the color is darker than the rest of the nucleus. You understand the reason? It's a little bit the same than what you said. So this piece of DNA contained in the genome are all located in the nucleus. And the structure, because this is what is interesting us, is the structure of the DNA that is transcribed by Pol1. So this bit is this famous 43 kb repeat, and you can imagine that there are plenties of copies of this fragment close together, and they can produce this 28S, 18S, and 5.8S. What is striking is that, I'm going just to talk to you about that, is that in this region, so you see the red boxes here with the little black arrow there. This is transcription start. So first thing that is quite complicated to understand is that we've got two promoters inside this repetition. So I'm going to talk to you first about what they call the 47S rRNA promoter and how it's working, and then I'm just going to tell you what this spacer promoter is. So technically, what is happening? RNA pol 2. So here at the bottom of the drawing, there is a zoom of this region. Okay, so you can see, for example, this part which is called T0 is this one here. The first 5' part that is transcribed is there, and then in red we've got the core promoter, and here it's not written, we've got these sequences. Okay, just there, in between there. So those two promoters, the promoter which is written 47S RNA promoter is the one that is recognized by POL1, and I will show you how they can interact, and then the transcription will give rise to the pre-rRNA that is going to be splitted in different versions of the ribosomal RNA. In front of this, so I will come back later on the notion of promoter. So if we look at the zoom, so here you've got the transcription initiation start, and what they call the core promoter, what we call in general the core promoter, is the part of the promoter that is going to be able to interact directly with RNA POL1. And the upstream sequence are the ones that are going to activate the RNA polymerase 1. So I just put this slide here just for your records, because after it's going to be very complicated and I will never you ask questions. So it means that if you want to know how sorry, I've got problem with my video. If you want to know how the 47S RNA that is going to give rise to the ribosome is processed, and I will show you later on. So first of all, we've got one single RNA, and then there are plenty of sites. It's going to be cut. There is a code here. So if you want, I can explain you that, but I won't ask you any question on this. It's just for your record. If you want to go back on how the rRNA are processed, you have this in reference. The only thing that we need to keep in mind is that the first RNA molecule that is produced is a single RNA molecule that contains all the precursors of the final 3 RNA that is composing the ribosome. And it's not necessary to remember all the steps because it doesn't make sense. But what you have to keep in mind that the processing, and we will come back on the processing of RNA later on, the processing of this pre-rRNA involve different steps, which are, there is endonucleotic cleavage. It means all the little black arrows are the position where the precursor RNA can be cut. So you see it can be cut in plenty of different positions. Then, so for example, from the first 47S molecule, the first process is to cut the little piece on the 3' end. So you see it's removed, and so on. Then there is another cut here that split the molecules in two, and so on and so on. So the processing of the pre-rRNA is a combination of endonucleotic cleavage, like the black arrows, and also the activity of exoRNAs, either 5' toward 3' or 3' toward 5', which can be found here, for example. For example, from here, there is this one enzyme that is going to remove the 3' part of this precursor of the 18S RNA, and so on and so on. You see there is also exonuclease activity that are required for this step. and also for the 5.8S. So just you have this figure, so it's just a synthesis of all the processing of the ribosomal RNA, and just keep in mind that the RNA processing is completely compulsory in order to have the functional 3 RNA that are going to be used for the biosynthesis of the ribosome. And you need to have endonuclease and exonuclease activity in order to process. So this is just for your record. So we go back to RNA pol 1. So pol 1, the promoter is like that. Start of transcription, a core region of the promoter that binds the RNA pol, and the more detailed view of this pre-initiation complex formation for pol 1 is this one. So the drawing is a little bit different, but it's the same. Here you find back again the transcribed region by pol 1. So in the middle, there is the 5.8S rRNA. This is the repeat, and here this is where the place where the core promoter is located. And what you see is that the pre-initiation complex, like for pol 2 and for pol 3, it's a complex of protein. Okay, so we've got the polymerase, okay, that's obvious. And then there are all these transcription factors that are necessary to bind to the core promoter. And we will study that a little bit more in detail for RNA pol 2. So this is to give you an overview of the topology of what's happening in a gene that is transcribed by pol 1. There is, so I didn't put the legend, but we will see it later on. So among all these proteins that are forming the pre-initiation complex, there is one that we will find back in other promoter. It's TBP. It is a protein that is TATA binding protein. So it's a protein, I will show you the 3D structure just after, that is able to specifically recognize the TATA box element that we can find in the promoter of pol 1 gene. I tell you that because there are some misunderstandings. Most, if not all, the genes that are transcribed by RNA polymerase 2, they contain the TATA box. But you will see later on that it's not always the case. For RNA pol 1, so the polymerase that is transcribing the ribosomal RNA, it also contains a TATA box. Okay, so this is a common feature between pol 1 promoter and pol 2 promoter is the presence of TATA box, and therefore the involvement of this protein, which is the TATA box binding protein. Is it okay? So this is just to show you, because after we can make a comparison between the structure of pol 1, pol 2, and pol 3 promoter, just to have an idea about the differences. Concerning, because, okay, I forgot to tell you something. So for the 3 polymerase, we're going to see, at least for pol 1 and pol 3, we're going to see how the transcription starts and how the transcription stops. We're going to see the same for pol 2, but also we're going to study the extension of transcription for pol 2. So for pol 1, initiation of transcription, very similar in terms of structure to what we can find for pol 2, because there is this TATA box and this TATA binding protein, but the rest of the transcription factor and the polymerase are completely different. Okay. What you can see here, I tell you just before, is that the transcription start, okay, is located after all the protein that interact with the promoter. Okay, just keep in mind this, because you will see it's not the case for pol 3. So for pol 1, the termination of transcription is very, very straightforward, and you will see it's much more complicated for pol 2. So here in this drawing, you've got the end of the DNA that is going to be transcribed by pol 1. So the promoter is on the left, 18S, 28S in the middle, 28S. And if you go on after the end of what is going to be the 28S, there is a sequence in the genes, okay? So in the DNA, there are sequences that are repeated from 1 to 10 times, okay? Depends on the repetition. And this is, take care because it's completely different for POL2. The sequence that are responsible for ending transcription by POL1 are at the end of the transcription region, and they are on the DNA, and there are proteins, so termination factor, that are recognizing directly the DNA to stop the polymerase activity. And this, we see it very clearly in this drawing. It means there you've got the polymerase, which is arriving at the end of the transcribed region, which is here. The little black line here is the new synthesized RNA, okay? And the polymerase is going to stop because at the end of the transcription unit, you've got protein-DNA interaction that blocks the polymerase, and then the polymerase stops, okay? So you see, for POL1, it's very, very simple. We've got a simple promoter that is recognized by general transcription factors, such as tata binding protein. All the initiation complex is constructed just before the initiation start. Once everything is there, the transcription by POL1 can start. So you've got the pre-ribosomal RNA, which is transcribed. Then the polymerase arrives till the end of the transcribed region. If there is always protein that are bound to these termination sequences, and the POL1 stops there and starts again the cycle. Okay, so you see POL1, it's quite simple. It's always on, and the ending of transcription is achieved through the interaction, the direct interaction between proteins and the genomic DNA. Is it okay for you? Okay. Now we're going to look at POL3. We're going to do exactly the same. How is the initiation and how is the termination? Because this is the only important point to compare between the three polymerases. So POL3 implicated in the biosynthesis, the transcription of plenty of small RNA, and it transcribes as well the 5S RNA, which is a ribosomal RNA as well. So for the ribosome, you've got 4 RNA. You've got the 28S, the 18S, the 5.8S. Those 3 ones are transcribed by POL1. And the last one, the 5S, is transcribed by POL3. Okay? So this is not the aim of the lecture to know which RNA are encoded by which polymerase, but I'm more interested in the complexes that are formed on the promoter. So for POL3, it's completely different. It's very, very atypical, let's say. So the pre-initiation complex is represented in this drawing. The example is, okay, so the target genomic DNA, there will be the RNA5S gene, so DNA, that are located in tandem. So a lot of copy of them in order to produce the 5S. So the topology of the promoter region, so around the initiation start, so this is this little black arrow, it's like this drawing. So there is one important feature is that the basal promoter, this is what is written at the bottom, the basal promoter elements, so it means the element present in the DNA sequence that are responsible for interacting with all the transcriptional machinery. They are localized downstream the transcription start. Okay, so do you see the difference between POL1 and POL2 that we are going to see just after, but you already know it. For POL1 and POL2, all the pre-initiation complex containing the polymerase, they are established on the +1 site, so transcription start, and the interaction are upstream the transcription start. In the case of Pol3, it's the opposite. The first protein, the two first proteins that are able to recognize a Pol3 promoter are TF3C and TF3A. So the blue and the green one on the drawing. So they are a little bit similar to the conceptual function of TATA binding protein. Okay, but they are not at all TATA binding protein. There is a TF3B, and you see it may contain TATA binding protein, but that is not at all used for recognizing the TATA box because there is no TATA box there. So sequentially, what's happening on a promoter that is transcribed by the Pol3 is that you have these two general transcription factors, we call that like this, TF3C and 3A, that recognize the sequence, which is the A box and EIE, okay? They are direct interaction. And it is once those two factors that have been bound to the DNA that all the rest of the proteic partner can interact. And you see the Pol3 comes after TF3C and TF3A. Okay. The only thing that is really peculiar, this is why I wanted to talk to you about that, is that it's the only polymerase where the formation of the complex, the pre-initiation complex, is happening downstream the transcription start. Well, it's not a problem. The polymerase start reading there, and the transcript will be the copy from the transcription start. Okay. Okay. So termination, I didn't put a slide for termination of Pol3 because I didn't find any information on that, and it's not really I didn't find any documentation sufficient for that. So we shift to Pol2. So we keep in mind. So do you have any question for Pol1 and Pol3? And from now, we are going to shift to Pol2.
I just one question for Pol3. So because it is downstream, the transcription start, right? So when the RNA pol want to come to pause the starting of the transcription, it needs the factor, right? The factor. So it's gonna be, I don't know how can it start it connects the iron bone with in the transcription start, but not like downstream with the promoter vessel like that. It had to make a hair clip or something to for them to connect to each other.
What do you mean? You mean because of the presence of those two one there? You're asking why it start there?
No, I'm asking like
I didn't get you.
So because this is in the basal promoter element are localized downstream, right?
Yes.
So it's not near the start at all. So when the RNA pol come, how can the basal promoter connect with the RNA pol if it's downstream? Because I know the pol need to start from the promoter start, right, and run down to all the gene.
Okay, I see. Okay. What you say makes completely sense. Actually, all the RNA polymerase, they don't start at +1. Okay, transcription start, I will call it +1. Okay, they don't. No, sorry. The transcription start at +1. This is the definition. Okay, we agree with that. But no polymerase recognize the +1. Okay, they don't know the +1. It is what makes the position of the +1 is the interaction of the first proteic factor that interact with DNA. Then they interact with the polymerase, and it is because, let's take an example. TF3C and TF3A, they have the capacity to recognize specific sequence in the promoter, okay? Even if it's downstream. So you have a piece of DNA with two proteins that are bound to this DNA, okay? From that, the Pol3 has a very strong affinity for TF3C and TF3A, so it goes to the interaction. And it is once Pol3 is there that it's going to start transcribing the first nucleotide at the catalytic site. You see what I mean? So the transcription start is defined by where these factors are interacting with DNA. It's not the polymerase that define. I don't know if it is the answer to your question.
Yes, but if no, just to be specific, more specific, like so the TF3C and the TF3A, right? It's downstream, so it's not gonna. So when the pole connect with the TF3C and TF3A, it's not gonna start at the arrow that you draw, or it will start at the arrow. Point.
Yeah. No, it will start directly as soon as. So first event, you have TF3C and TF3A that are recognizing the promoter. From this interaction, you have all the other proteins of the complex that are going to be complexed. So Pol3 plus the other factors, okay? And once this mega complex is present, the transcription starts at this place.
Oh, okay.
Okay? It's the thing that you have to keep in mind. It is the first protein that interact with promoter. We're going to see it for Pol2. The first protein, for example, for Pol2, it's TATA binding protein. This protein is this one that is going to put the RNA Pol2 at one specific place. And the Pol2 does not recognize DNA. So once it's there, it's going to start. So technically, it is never the polymerase that defines the +1. It is the first factors that recognize DNA that is going to put the polymerase at the right place to start at the right place. It's okay or what?
Yes, it's okay.
After, there are in biology, there are so many exceptions. For Pol2, we're going to see it. I explain you after. We can have different transcription start for a single gene. So that's another point. So we go for Pol2, and I will make more comments about this on this transcription start. So the Pol2 promoters. So this is really, and we're going to spend two full lecture on Pol2 promoters and interaction. So this is the typical topology of a polymerase II promoter. So what you can find, so you have explanation there. Actually, it's quite interesting. So what's happening? We've got in the center, because it's always all the promoter study that we do, we use as a reference the transcription start, because it's from there that we can say it's +10, -20, and so on. So the reference in terms of position is the transcription start. So this is what we call the initiator. So this will be the first nucleotide that is going to be transcribed. Okay, we always have, for every, but not always, most of the time, this is what I was telling you, 30 base before the transcription start, we find this famous TATA box that is going to be able to be bound by the TATA box binding protein. And then there is also another element that is directly linked to TATA box. It's the BRE, which is the site for recognition of TF2B. So this is the two first factors that need to be bound to DNA in order to recruit RNA pol. So here you've got the sequence, the consensus sequence of these boxes. One point, I'll show you a graph after that on that. It's very complicated to analyze promoter sequences because you see it's very short sequence. So for example, TATA box is delimited by 6 nucleotides. And if you check in the whole genome, all the sequences that are TATAA, you find plenty, and they are not necessarily data boxes. Okay. After, if you look at this part of the drawing, this is a sequence analysis of the sequence located at the +1 site in Drosophila, so in the fly, and in human. Okay. So there is no similarity at all. So the transcription start is not something that is canonical. This is canonical because this is present in most of the genes, but transcription start sequences are not conserved at all. So this is the consensus sequence, for example, for all the genes in Drosophila. So you see that at -2, there is always a T, -1, always a C. The transcription start in Drosophila is always A, but the +2 is either G or T, and so on. So in Drosophila, you can say it's quite obvious. If you have TCA and a data box 30 base before, you should get a POL2 promoter. If you look at human, the consensus sequence of the initiation start is not that clear. It's still an A for the first nucleotide, but then after you see that before -1 and -2, it can be a pyrimidine, whatever pyrimidine it is, and so on and so on. So what you have to keep in mind for that is that if you look at these little boxes arranged, so look at the position. Here in the middle, we are at the transcription start. There are very important sequence just a little bit before the transcription starts, so the data box and this TF2B binding site, very important for polymerase activity. And just a little bit downstream, it's not very far, it's also around 30 base after the +1. There is sometimes this kind of motif that some of them are conserved and some of them are not conserved at all. And if you look at the text there, just to show you that it is not always the same. They explain you that in many genes that are transcribed at low rate, okay, they call that a housekeeping gene. They do not contain the data box or an initiator. Okay, they contain instead of GC-rich region and something like that. So what you have to keep in mind is that even though most of the genes transcribed by POL2 contain the data box, there are plenty. It's not very rare that in which you will never be able to find this kind of sequence, consensus sequence. Okay. So this is the structure because I had to talk to you about the structure. Then what is happening during the pre-initiation complex formation? So during initiation, so this is a very short summary of the promoter, the core promoter of POL2 gene, the data box, and as you see just before, the first event, it's exactly like POL3. Yeah? The first event, it's not the polymerase that binds the promoter. This is transcription factor, general transcription factor. So data binding protein that bind the data box and the TF2D that we saw before that is able to recognize the sequence before or after the data box. Because when we talk about the orientation in promoter, it's very, very strange. So first event, to make it short, first event, recognition by general transcription factor or of common promoter feature. Then you have a succession of new proteins that are interacting, and it's only once we've got this basal complex that is linked to DNA that the polymerase, the real polymerase, the one that is carrying the activity of polymerase, can join the complex. Okay? And once the complex is joined, the system is not active. So you've got all the transcription factor that recognize the POL2 promoter, the RNA polymerase that arrives by interacting with this promoter, with this protein, but it's not working. And the start of the polymerization has to be done by phosphorylation. You see this blue tail here. I don't know if you see my mouse on the screen. It's the C-terminal domain of one of the subunit of RNA pol2. That is very important. I will show you after that. And it's the place, this C-terminal part of the RNA pol2 is a part of the protein that is going to interact with plenty of protein, other transcription factor, I will show you just after. And it needs to be phosphorylated to have the RNA polymerase activity. So 3 steps. The first steps are direct recognition of general transcription factor. After this step, once the general transcription factor are bound to DNA, pol2 can join the big complex. And last step, to start transcription, you need to phosphorylate the C-terminal tail of polymerase II. And here I just put you a schematic view of the interaction between TATA binding protein and TATA box, just to show you that it is not pol2 that interact with DNA. And you see, actually, it's a dimer, the TATA box binding protein, and it directly interacts with the TATA box. This interaction is in the minor groove of DNA, not the major groove. Okay. So technically, we can say that it's TATA binding protein that is the first event in recognizing the core promoter sequence for pol2 genes. Okay. Do not hesitate if you have questions. So this is, I'm sure that you already studied that last years, but at least you have everything for reference. So then the cycle of polymerase for pol2. We have to think in a complete dynamic way. So the drawing here show you the whole cycle, and you have to imagine that it's cycling and cycling again, of the transcription of one pol2 gene. So at the bottom of the drawing, you've got the genomic DNA with the +1, so the transcription start. Okay, and the cycle. So CTD means C-terminal domain of polymerase II. Okay, so this is the site of phosphorylation, and the phosphorylation, they are those little red balls with numbers. Don't pay attention to the number. TF2H is another general transcription factor that is very important, and I will explain you why. So the cycle first. The promoter has been recognized by TATA binding protein. So the polymerase, which is not phosphorylated, can join the complex. Then, at that stage, we've got everything to start transcription, but no transcription activity because the C-terminal domain is not phosphorylated. Okay? So everything is ready to start, but no transcription. One last event is that there is a transcription factor, a general transcription factor, named TF2H. And this transcription factor, it carries 2 enzymatic activities. It is a kinase, so it's going to be able to phosphorylate the C-terminal domain of pol2, and it contains also an activity of helicase. Do you know what it is? Helicase, H-E-L-I-C-A-S-E-E.
It cuts the helix
Yes.
of DNA.
It unfolds the helix.
Unfold.
It doesn't cut that directly. So this TF2H has a double function, which is extremely important. First, he is able to phosphorylate the pol2, so the polymerase will be active. But in order to do the transcription, the polymerase needs to have the two strands of DNA that are open, and this is done by the helicase activity. So the helicase activity, it takes the double helix and simply unfold it to be able to be read. So once this TF2H arrive at the pre-initiation complex assembly, okay, the pole 2 is phosphorylated, and you see that the RNA is produced. It goes on, so transcription goes on. Pay attention to this. The mRNA already has a cap while it is not transcribed fully yet. Okay, it's not finished, the RNA, but it has already a cap. We will come back on this cap after, okay, because it's RNA edition. So pole 2 inactive, join the promoter, the core promoter, because of the Tata box binding protein. TF2H phosphorylate and unfold DNA, transcription occurs, and at the end, and you will see how it's happened because I prepared the slide for that. Once the polymerase arrives to the termination, the stop of transcription, which is complicated for pole 2, the polymerase is not linked to DNA anymore, so it goes out of the matrix. It is unphosphorylated by a phosphatase, so you don't need to remember the name. FCP, P stands for phosphatase. Okay, so all the phosphate groups will be removed from the C-terminal domain, and the pole 2 is inactive again and can enter back a cycle, and we go on. Okay. So just to show you the importance of this C-terminal tail of the pole 2. So the CDT, it's a neptapeptide, so 7 amino acids, okay, which is repeated a large number of times, so between 27 and 52 times, okay, depending on the species. So this heptapeptide is always YSSPTSSSSPS. And serine residue are the amino acids that can be very easily phosphorylated. So the phosphorylation of the C-terminal tail of pole 2 occurs at the serine residue of this repeated heptapeptide. Okay, so this is just for information. Okay, so keep in mind that one complex of polymerase transcribe a gene once and go on again, and it goes again, and it goes again. So now we're going to see, do you have any question on that? After we're going to see how we study the promoter and all the transcription factor, but I needed to explain you how the pre-initiation complex was established and how it works. Is it okay, or do you have any questions? It's okay? Whoa, yes, somebody is coming just right now. 1 second. But did you add the information that we start at 8 today? Yes? Okay, well, no problem. So we're going to see something that is very general, but always for pole 2. We're going to see what we call its extremely wide domain. This is all the world of RNA. And I just want to talk to you because now, for now, it will be okay for you, what we consider for the RNA processing. So it's a very important part of the life of an RNA, because as soon as an RNA is transcribed, you need to have plenty of different modifications to carry the function. So for the polymerase 2, you all know that mRNA has a cap in the 5' end, a poly-A tail at the 3' end, and you all know that the genes in eukaryote contain introns and exons, so you know about splicing. After, I don't know if you already heard about that, RNA edition, which is the chemical modification of bases. Have you ever heard about that or not?
No.
Okay.
Nothing.
Oh, so I will show you just one example. So it's just to remind you what you already had in your lecture. So we're going to go very quickly on CAP, very quickly on polyadenation. just a quick update on splicing, and I will explain you just 2 or 3 things in terms of RNA addition. And after that, we're going to see how technically we study protein interaction in the promoter region. So just for the capping. So the idea is that you understand by the 3 or 4 following slides that all the process happen at the same time during transcription. It's not a sequential process. So you have, this is on the left, you don't need to know that, but you have it as a reference. This is the chemical modification of the 5' end of RNA that is just transcribed from Pol II. So the 5' is always the phosphorylated part of a nucleic acid. There are a succession of 3 or 4 steps. Basically, what you have to keep in mind is from the 5' extremity, which is a phosphate extremity, at the end you have a guanosine that is linked in a very specific way because it's GPPP. So it's linked reversed than the normal nucleotide, and it's methylated, moreover. So this is what we call the cap of the RNA. So every RNA that is synthesized by Pol II has a cap. The function of this cap is mainly to stabilize RNA during the export into the cytosol before being translated. They can be also involved in regulation of translation, but okay, it's a little bit more complicated. So what I wanted to show you is not to talk to you directly about the cap, because the cap is just a chemical modification at the 5' end of the messenger, but it is what's happening in the context of the RNA polymerase complex. So here what you have, this drawing is, in gray is the genomic DNA, in blue, it's the polymerase. You see the C-terminal domain of the polymerase. You see it's phosphorylated. So on the top of this drawing, we are in a situation where the polymerase arrived to the core promoter. It was phosphorylated and it's already transcribing. So here we are in the middle of transcription. Okay, so the polymerase is starting to scan the transcribed region of DNA. And you see that this little blue line is the RNA, which is produced little by little as the polymerase is moving. And what's happening, I think it's a very good summary, this drawing, even if it's completely schematic. This C-terminal tail, okay, needs to be phosphorylated to have RNA polymerase active, but it's also the site of interaction of plenty of other factors. And look at the names of those factors. They are the capping factors, the splicing factors, and the polyadenylation factors. So it means that as soon as the polymerase starts its job, which is synthesizing the DNA, polymerase can be active only if it's phosphorylated. Okay, fair enough. But the C-terminal tail of polymerase, once it's phosphorylated, can interact with plenty of different complexes that will be implicated in the processing of the new RNA, which is information. So the cap, this chemical structure there, needs to be added at the first nucleotide that are produced after the very beginning of transcription. The capping factor, they are already present just at the right place. So what's happening is as soon as the new native RNA that is coming out the polymerase, those capping factor will interact with the 5' end of the nascent RNA, and all those factors, they will ensure the removal of the phosphate, the transfer of the guanyl, and the methylation of the rest. So this is a complex of factors. Okay? It will be the same for splicing, and it will be the same for polyadenylation. So it means that when a mRNA, a pre-mRNA, is transcribed by Pol II, the first introns that are produced are going to be removed even if the total length of the RNA is not synthesized yet. So all those processes occur at the same time. It's okay for you? So the polyadenylation. So we start just the capping, 5', the other end, the polyadenylation. So you see, we start for exactly the same thing, and I'm going to show you, so I need to explain you two key features in this slide. First one, the polymerase arrives, so it's synthesizing RNA, it arrives at the end. I'm going to talk to that just after. The C-terminal tail of the polymerase carries the splicing and the polyadenylation factor that we just saw in the slide before. Once the RNA is stopped to be transcribed, you have these polyadenylation factors that are going to be. Do you remember for POL1 transcription termination? The POL1 transcription termination was obtained because some protein were directly interacting with a DNA sequence at the termination site, and so the POL1 has to stop. For POL2, it's completely different. So we're going to see, it's a little bit complicated because everything is linked together. In gray, and with the little orange spot here, this is the genomic sequence. Here, we've got a sequence that is going to be, but not directly. This is the important point I want to talk to you. It's going to be implicated in the cleavage and the poly-R of the RNA. It is encoded in the DNA, but the action of this DNA sequence is not happening at the DNA level. I'm explaining myself. The polymerase is polymerizing. It arrives to this sequence. Look, the next step, the polymerase go on transcribing this little orange sequence. So this sequence that is contained in DNA is going to be found back in the RNA because it has been transcribed, okay? And it's once this sequence in the DNA has been converted by POL2 to RNA copy that the modifying factors can interact with RNA in that case and not with DNA like POL1. Interact with this sequence, and the consequence of this interaction is extremely simple. It's double. It cuts the DNA, it stops, and then it recruits another protein which is poly-A polymerase, PAP, which is an enzyme that is going to add AAA at the end. Okay, so as a summary, the polymerase 2 transcribed one sequence that is found back in the RNA, and it is this sequence in the RNA that will give the place of stopping, which is cutting the RNA there, linking to the poly-A polymerase, so the poly-A tail can increase, and as soon as the poly-A tail is produced, it's directly surrounded by the interaction with a protein, which is a poly-A binding protein. Okay, so all the poly-A tails is completely coated by a protein that has a very high affinity with poly-A RNA. So do you see the difference between the termination of POL1 and the termination of POL2? Conceptually, that is completely different. Do you understand? Okay, so remember, this is why I show you POL1 before. POL1 stops because of a DNA sequence that is recognized by a protein. POL2 stops because it transcribes a DNA sequence in RNA, and it is the RNA sequence that is going to be responsible for the stop of the transcription and the subsequent addition of the poly-A tail. Okay, so it was just to be sure that, so you see there are common features between this polymerase and some very specific feature of the polymerase. One point on the poly-A tail, the size here, they show you 200. The size of the poly-A tail is very, very different from some messenger to other messenger. And there is, but we won't talk about that in this lecture, but I think it's important that you have that in mind, is that the size of the poly-A tail is directly proportional to the half-life of the RNA in terms of number of translation. What I want to say is that, for example, one RNA that contains a very long poly-A tail is going to be able to be translated much more time than an RNA that has a small poly-A tail. Because we won't talk about translation in BP10, because during translation by the ribosomes, every cycle of transcription, there is a shortage of the poly-A tail. So basically, you can say that the size of the poly-A tail will be more or less proportional to the number of cycles of translation. Okay, so poly-A tail, it has multiple use. It stabilizes the RNA, okay, but also it's implicated in the number, in the quantity of translation of the' but we won't study that here. So let's go back to the RNA processes. We saw the cap in the 5' end, specific to POL2, the poly-A tail and the termination on the 3' end, and this is just a quick, because after I'm going to explain you what they are, what's happening for splicing. So do you know what is hnRNA?
I remember you wrote before that it is heterogeneous nuclear RNA.
Yes, heterogeneous nuclear RNA. So what are they? Because all the researchers, they give different names to RNA, but sometimes they are the same. hnRNA, they are what we used to call the pre-mRNA, which is the mRNA that is in the nucleus and that has not been spliced. In other words, it's the full-length genomic DNA, exon plus introns, that have been transcribed. Okay, but the introns haven't been removed yet. This is what they call heterogeneous RNA. So it's pre-mRNA. So this is just the cycle, the different process that happens to remove an intronic sequence located in between two exons. Very important in between two exons. I explain you one thing. So I'll show you after when I'm doing. So an intron is defined by being surrounded by two exons. That is the definition. So we've got two different sites that has to be removed. I'm not going to explain you all the process because you already studied it. I just want to show you something. So there is no specific sequence there. There is a common feature inside the intron, which is a consensus sequence that can be recognized by specific splicing factor that recruit different proteins. And look, I just want to focus on that. They are called SNRNP. It means small nuclear for SN. RNP is ribonucleotide protein. So it means U1 and U2, they are big complexes that are containing proteins and RNA as well, like ribosome. Ribosome contains protein and RNA, but those splicing factors, they also contain RNA and protein. According to you, why do they contain RNA? What can be the rationale?
To find the specific site for to cut.
Yeah, there is two aspects. This one you are completely right. So the specificity of the site that is recognized. And also, do you know, because the word of nucleic acid is completely wonderful, do you know that some nucleic acid can be catalytic as well? Have you ever heard the term ribosome?
Yes, the ribosome that can have the enzyme function.
Exactly, but U1 and U2, they are very similar to, conceptually, they are similar to ribosome. They are composed by protein and RNA, and they've got an enzymatic catalytic function, which is done by the protein part and the nucleic acid part. So this is, in terms of mode of action, it's very similar to what's happening in a ribosome. And the consequence won't be protein polymerization, but they will cut the RNA. So you have all the process. It implies a loop and so on and so on. It is not the problem. So it was just to show you, because I'm going to explain you a little bit this part, which is containing RNA, where this RNA is coming from. Anyway, so this is the process of splicing one intron, and what you have to keep in mind is that an RNA inside the cell is never naked. It's always surrounded by plenty of protein in order to protect it. So this drawing here is completely the schematic of what can be an RNA inside the nucleus just after transcription. So the transcription is finished. You see we've got the cap, the first exon, the junction exon intron here, U1 is here. This is going to be removed by excision of the intron. Always U1 on the left, U2 on the right. Then the second exon, U1 on the left, U2 on the right. This is the last exon, and the poly-A binding protein at the end. So in the nucleus, HN RNA is in this type of complex. And the bottom figure there is just to show you what could be the consensus sequence in order to identify intron-exon junction. You see it's not very clear. The sequence that is recognized by U1 is quite well defined. Okay, we say that we know that it's A, G, and then there is a consensus sequence in the intron. That's fine. If you look at where U2, and if you check very well, U2 doesn't recognize the other junction. It recognizes inside the intron. U2 will recognize this part, and this is this famous A, which is red here, okay? The consensus is not very clear, but because, look at the mechanism, because of the binding of U2 at that place, the excision will occur at X nucleotide away from that site. So this is just, so you see, when you see this consensus sequence, you can say, okay, now I can check in all my genes if I found A, G, and so on, and you can say it's going to be exon-intron junction. Take really care about that, because it's not always the case. It's completely almost impossible to predict the site of splicing based only in sequence analysis. There are some algorithms that are quite efficient now, but it's very risky. Okay. So just to come back on this U1 and U2. So I found this quite old, but very interesting. I mean, it's a bad copy. There are two parts, the left and the right. So it allows me to introduce these two two other classes of RNA, which is snRNA and snORNA. It means small nuclear RNA and small nucleolar RNA. Okay, that's all. These are two small RNA, and just want to show you this part and this part. We can check both of them. So the snRNA processing, I want that we start here, and I just want to pay attention which polymerase is transcribing this snRNA gene. You see it's polymerase 2. Is it okay? What you probably heard is that all the genes transcribed by polymerase 2 can be translated. But there are few exceptions, like this type of genes. The SN genes are transcribed by polymerase 2, but will never be translated because their function is not carried by the protein, it's carried by the RNA itself. So this snRNA are transcribed by Pol2 in the nucleus, exported in the cytosol. There are a few modifications, okay, cap methylation and so on, re-imported back in the nuclei. They are present in some subnucleus body, which is called, you heard about this term, causal body already? No?
No.
Okay, but because inside the nucleus, using microscopy, there are plenty of small spots, and a lot of researchers gave them some names. And there is a very famous subnucleus structure, which is called the causal bodies. Okay, and actually, this is placed in the nucleus where you got a huge amount of this snRNA specific RNA and protein. So technically, we have genes in the genome that are transcribed by Pol2. The RNA is exported in the cytosol, come back to the nucleus. Those RNA interact with some snRNA specific protein, and this nucleoprotein are the U1, as an example, U1 and U2 complexes that are implicated in splicing. So I think it's important to understand because we have, if we take the example of a classic Pol2 gene, so a gene that contains exon and introns, okay, that is going to be transcribed, the introns are going to be spliced, the messenger RNA is going to be translated into a protein. So this is the classical case of a Pol2 gene. But you have to keep in mind that in order to have the step of splicing that is happening, you need other genes also transcribed by Pol2 that are going to give rise to snRNA that are going to be part of the complexes that are responsible for splicing. In other words, if a classical Pol2 gene needs to be spliced at one moment or another, and to be spliced, some other genes also transcribed by Pol2 need to be active. You understand what I mean? So it's quite tricky because you need Pol2 in order to have Pol2 functions. And just a short explanation for the snORNA, so the small nucleolus RNA. It's exactly the same. They are implicated in the ribosome synthesis. Okay. For the SN, it's the splicing factor. For the SNO, it's the ribosome biosynthesis. It's conceptually exactly the same. It is those SNO genes are transcribed by Pol2, exactly like the SN genes. The only difference is that the pre-SNO RNA doesn't go in the cytoplasm before coming back to the nucleus. The pre-SNO RNA stays in the nucleus and is interacting with protein and then go directly into the ribosome biosynthesis. So I put this slide just to show you one thing that can be a little bit problematic to understand, is that conceptually you have some partners that are necessary in order to have RNA processing done, that need also to be transcribed from the same polymerase than the target one. Okay, that's the point. Do you have any question on this? Or is it okay? Let me check. We make a small break. Is it okay?
Yes.
Okay. So it's 35. We start back in 10 minutes. Is it okay for you? Okay.
Yes.
Okay. So you tell me when you're okay to go on. Can we start again? Okay, let's go. I didn't see you. So, so this is from this schematic. This is the idea of splicing, but there is something, and we will see that it's really more and more well characterized, that they are in terms of function, because this is what is interesting us. There is a process in the RNA which is extremely important, which is RNA alternative splicing. So the normal splicing is you know it very well. I mean, you remove the introns, and the alternative splicing is a way to, from a single gene in the genome that is going to be transcribed in one single HN RNA, from the single version of the molecule of the HN RNA, you can produce different type of protein, because some exon or some intron are not removed in the canonical way, let's say. So this example, it's coming from a paper. The reference of the paper is on the next slide. It was studied in plant. So I won't explain you all the details of this figure, but you will understand perfectly. So it's this paper was a study on a gene that is in plants. It's coming from plant. It's called APX. APX stands for ascorbate peroxidase. So it's an enzyme that is implicated in detoxifying oxidative stress in plant. It's very important. And there is only one gene that exists. The gene exists only in one copy. So I'm not going to explain you, because they use a transgenic approach in order to prove the alternative splicing. So in the part A of the figure, you see the construct of the transgene, but I want that we look at the part of the endogenous chloroplastic ascorbate peroxidase. So this gene, this is only the end of the gene, because you see the number. It's composed with plenty of exons and plenty of introns. So you see there is at least 13 exons. And this is intron number 12, this is number 11, number 12, the little one here, and so on and so on. So what is happening is that once this gene is transcribed by Pol2, so it's a classical Pol2 gene, the pre-mRNA, so the hnRNA, is composed by all the introns and all the exons. And once the splicing occurs, there are two different versions, and actually there are more versions, two main versions of the processed RNA that are produced. So there is one version, and I will show you just after what's happening. The first version is the normally spliced version of the hnRNA, which means that exon 1 is conserved in the mRNA, intron 10 removed, exon 1 conserved, intron 11 removed, exon 12 conserved, intron 12 removed, and conserved intron 13. So from that, you have one protein that is going to be translated that contains the coding sequence of exon 10, 11, 12, and 13. But in other parts of the plant, in other organs, and I will show you how, what is the regulator of this, you find an alternative version of the splicing of this hnRNA. And this alternative version contains exon 10. Intron 10 has been removed as it is. Then just after exon 10, you find exon 11, the coding sequence corresponding to exon 11. And then directly after the coding, the protein sequence ending up at exon 11 is directly going on exon 13, which means that in this alternative splicing event, instead of removing intron 11, keeping the 12 and removing intron number 12, the whole block containing intron 11 to intron 12, which means including the exon 12, everything is removed. And they call that intron 12 in that case. So the translated protein from this messenger RNA that has been alternatively spliced will be the coding sequence of 10, 11, and then directly the 13. So it's a very interesting way for a living organism to, from one single gene, to be able to produce two different proteins. Moreover, you can imagine that those two versions of the protein can be produced in different places. Do you follow me for that? I don't see what you are thinking. Is it okay for this alternative splicing?
Yes. This also happened in a human, right?
Yes.
Not just only in
No. It happens in all eukaryotes. In Saccharomyces, it hasn't been reported as far as I know, but Saccharomyces gene structure, there is very, very few introns. So you understand the concept. It's very simple. You've got a kind of possibilities mixing different exons, always in the same sequence, but you can remove some of them because they are considered as introns. But so what I was saying is that from this kind of concept, you can produce two different proteins in two different places, for example. So what would be how can I say, the model, the conceptual model that could explain that you can produce two different proteins in two different places coming from one single gene? Okay, we'll see. We're going to see the next slide, and we're going to talk about that. So there is something. So you understand the alternative splicing process, but if we look carefully the sequence, so the authors of this paper, they analyzed the sequence, and they wanted to study this sequence, which they name SRE. So you've got the sequence here, okay? So don't pay attention to the mutation because it's part of their research. So the normal, the endogenous sequence is this one, the last one. So there is a small sequence which is partly on the exon 12 and partly in the intron 12 in the gene that you find back in the RNA. And this sequence is the regulatory sequence of the alternative splicing. So I show you what they did just after. So here we've got a kind of, so this is the real experiment. We are going to come back in this type of experiment later on for promoter studies. So what they did is, are you familiar with the technology of electromobility shift assay? EMSA, gel shift. Have you ever heard about that?
It is similar with the electrophoresis or not?
It is an electrophoresis. Technically, it is an electrophoresis, but it's called electromobility shift assay. It's a way to analyze protein DNA or protein nucleic acid interactions. Do you know a little bit about that? Anyway, in a few slides, we will use a lot of this kind of technique. And on Monday, we will have an exercise where we'll use a lot of this technique. So I just change, I shared, change something, and I will just show you conceptually how it works. It doesn't work. Okay. You see the whiteboard? It's okay? Just tell me if you see the white. Okay. So, so it's called, you will find it written like EMSA. It means electromobility. E is for shift. Mobility is the M. Sorry. Electromobility shift assay. Okay. So what is done? So for example, they take the concept of the experiment. You have a piece of DNA or RNA. Okay. Anyway, nucleic acid. Okay, so in our case, for the alternative splicing, we are checking for RNA. So this fragment of RNA, in our case, and after you will see we only use DNA, it is labeled with radioactive phosphate. So you label it with radioactive phosphate. Then what is doing, the experiment is like that. You have one first tube and a second one. In the first one, so this fragment of RNA, we call it the probe. Okay? So first experiment, you put only the probe. Okay? Second experiment, you put also the probe. Then you add protein extract. In that case, we don't add. In this case, we add protein extract. Okay? You tell me if you don't understand. So two situations. One, where we put a piece of nucleic acid that has been radioactively labeled. And the second one, we put the same piece of DNA labeled and protein. So if in the protein extract, there is protein that are able to physically interact with the piece of DNA or RNA, it will form a complex. So if this tube is tube number 1 and this one is tube number 2, in tube number 1, we will find only the piece of DNA with the label on both ends. And in the second tube, if there is an interaction, we will find a protein that is bound to the nucleic acid. Is it okay for that?
Yes.
Okay. Those two tubes, we use electrophoresis, but it's a kind of special electrophoresis. It's done on polyacrylamide, okay, like proteins, but very low percentage. It's generally 4%. Normally for protein electrophoresis, it's around 10%, so it's not a very strong gel. You put the two samples on the top, and what is going to happen? So we put the electricity. If we put sample number 1 and sample number 2 in those two things, so first tube, you only have the probe which is double. The DNA fragment will mutate, and because it's radioactive, you will see, if you do an autoradiography, you will see a big dark spot, and this is the size of what we call the free probe. We say free probe because free, it's completely naked, there is no protein on the top of that, and this lane of the electrophoresis is just to show you what is the distance of migration of the bit of nucleic acid. Okay? If in the tube number 2, in which we put the protein plus the probe, if there are some protein in this mix of protein that interacts with the DNA, they will form a complex, protein plus DNA, or RNA in our case, that will have a really much higher molecular weight because of the size of the protein. So what we are going to see, because we put a lot of probe, we always see the free probe that is not bound, but we will see the formation of a complex that is bigger. So the migration occurs in that direction. So the smaller molecule are the bottom, here, smaller molecule, and complexes on the top. Okay? So this we call the electromobility shift, because there is a shift in the mobility because of the presence of the protein. Do you understand the concept of this? Yes or no?
Yeah.
So technically, the idea is very simple, is if there is protein nucleic acid interaction, the formation of the complex will give rise to higher molecular weight complex, and the consequence of the interaction will be less migration inside the gel because of the size of the complex. And this is why we call that electromobility shift assay. Technically, I will show you after, because we can do and especially on Monday, extremely powerful things with this very simple technique. So I shift back to the PowerPoint presentation to explain you what they did in the context of this alternative splicing. So what they did at the bottom of this of the slide, you've got once again the same structure of the gene, the one that we saw before. So here there is this famous SRE sequence that they study. You see it's in between exon 12 and exon 13. Okay, this is the new intron when there is alternative splicing. So what these researchers, what they did is they took this sequence from the RNA. So they produced it synthetically. Okay, so they've got this piece of RNA. And this is the electromobility shift assay. So there are 6 lines. Okay. There are plenty of redundant lines. So we're going to see first the first lane on the left. It's a lane, so you see what they put in the tube. In the tube, they always, in every of the 6 lanes, they put the probe. The probe is this fragment of RNA which is radioactively labeled. Okay? So all the tube contain the RNA fragment. And then they add or no different protein extract to this tube. The first one is always like that. It's always a control. You don't put any protein. This first lane is compulsory because it's with this data that you can say, okay, the free probe, which means the free RNA, the RNA which is naked without any interaction with protein, migrate there. Okay, that's your control. And then what they did, so they repeated the experiment, but we're going to look at only the two last lanes on the right. They used protein extract coming from nucleus. Okay, so this is protein extract. They obtained this extract from tobacco plants, and they used two different organs for the extraction. L means leaves. So this is, in that lane, they put the probe plus protein extracted from the leaves of tobacco. In the last lane on the right, they put the probe as well, but the protein they put are coming from the same plant, but from the roots of the plant. And here they do the same using spinach. Leaves, L is for leaves, R is from root, and S is for stems. Okay. So how can you analyze this type of figure? So we know the size of migration of the free probe, and what we see is, I put the red one, and what we see, if we use extract, protein extract coming from leaves, either from spinach or from tobacco, both of them, okay? You see that the probe is still here in all the lanes. It's normal because we put a lot of probe. It's always in excess. So we see the probe, but we see with a smaller distance of migration, so it's a mobility shift, we see the formation of a complex. So this dark signal here, which is above the free probe, indicates that in this extract, in this protein extract coming from leaves, there are protein factors that are able to interact with the free probe. And you see the complex which is formed. What you can see from this experiment is that if you do the same, but using protein coming from the roots, so another organ, if we worked on animals, we could have taken the liver, the heart, or whatever, different organ. You see that in the protein extract coming from the roots, either from spinach or from tobacco, we don't see the formation of the complexes. Okay? So what kind of conclusion can you draw from that? I'll leave you, give me an idea. How would you analyze this type of data?
Yeah, so from this result, I think that we can see that there is some elements and proteins or something that binds to...
No. You're sure that it is protein?
Okay.
Yes.
Yes. So I can see that there is no protein complex that can bind to this SRE RNA in the root tissue, but there is some certain kind of protein in the leaves and the stem. Therefore, there exists an element that can affect the tissue-specific alternative splicing.
Exactly. You said plenty of very important points. You said tissue-specific, and you are completely right. Then after this experiment, give you the information that this, and you said it, you're right, this RNA sequence, the SRE, is able to develop protein interaction, okay? And this protein interaction, we only see them in leaves. We don't have the formation of the complex when the extract is coming from the roots. So you're completely right when you say it's tissue specific. So if you look at the little drawing at the bottom, it means that when we see the complex formation here, it means that we are in a situation in which there is a factor that we don't know, it's a protein or a protein complex, that is able to bind this sequence. And what you see here is that this, they call that factor X. This factor X is present in leaves, but not in roots. Do you agree? So now look at the model. The model of the splicing, of the alternative splicing, is if you don't have in the organ, if you don't have factor X, the splicing will be normal, which is exon 10, exon 11, exon 12, exon 13. When factor X is present, factor X bind to the SRE element and change the splicing pattern. And then when factor X is present, the splicing will be exon 10, exon 11, and directly exon 13. Okay? So, if you have that in mind, what is the form of space of the spliced mRNA in leaves, and what is the form of the spliced RNA in roots? Okay, in other words, the mRNA in roots, does it contain exon 12, and the mRNA in leaves, does it contain exon 12?
The one in roots have exon 12, but the ones in leaf don't.
Exactly. So you see that everybody understood what she said. So in the roots, because you don't have the formation of the complex, it means that you will have exon 12, but in the leaves, you don't have exon 12 in the final protein. So, and according to you, why do we have this result for the stems? Remember, it's in spinac. It's a little bit tricky what I'm asking you. What is? You see that there is a complex that is formed in stems. Do you see it?
Yes.
But not as strong as in leaves, but it exists. It's different from root. So, from this, look, tell me.
Is this because the stem is between the leaf and the root, so it's like a transition from the entire tissue to the entire tissue?
No, nice try. No, no, it's not that. So it's not a tree, it's spinac, okay, or tobacco. What is the common point between leaves and stem compared to root? It's a little bit difficult what I am asking.
It has
think about
the chloroplast.
The chloroplast and the chlorophyll. You're right. They are photosynthetic. Okay? And this enzyme, we are talking about ascorbat peroxidase. Ascorbat peroxidase, I was telling you that it's implicated in detoxification of oxidative stress. And oxidative stress happens mainly during photosynthesis in the chloroplast. So you need to have much more detoxification processes in green parts of the plant than in the roots, which are not green at all because they don't contain any chloroplast. So actually, basically, it means one thing. If you want to go, but you couldn't know that. So if we go back to the very base of the experiment, you understand, I hope, that using this very simple method, you can check for protein nucleic acid interaction, and you see that here in the case of alternative splicing, from one single gene in the genome, you can have two variants of the protein in different places, okay? So in that case, we've got a different protein in chlorophyll in green parts, green organs, than the one that we found in the roots. After, I don't have any more information on these two variants, but one can imagine that the variant that is produced in the green parts is much, much more active in terms of oxidative stress detoxification than the one in red. But anyway, do you understand that with this kind of system, the cell can produce different type of function with a single gene? And moreover, as you said, it's tissue specific. Okay. Did you understand this idea? So imagine what can be done through alternative splicing. It's very, very powerful because you can have much more function than the number of genes by themselves, and in a tissue specific manner or inducible manner. Is it okay? I'm just checking the time. So we will finish in 3/4 of an hour because after I've got another lecture in France. Is it okay for splicing and alternative splicing? Okay. So I will finish very quickly with something you told me that you didn't hear before. It is what we call, it's the last final modification that we can find post-transcriptionally for RNA. It is what we call RNA edition. So it's something, it's another word, because you have to imagine that, so you know from the beginning that RNA is composed by 4 bases, A, U, G, C. But actually there are much more than 4 bases. There are almost 20 different modifications. So just to show you, but you don't need to remember that. For example, a cytosine can be deaminated, and it turns to be a uracil. So you can convert a C to a U, just chemically, okay? It's not during transcription, it's chemical transformation of the base. So it's just to show you what it is. Just want to show you, it's a little bit disconnected from the rest. I extracted 2 examples of RNA edition. Once that is happening, and it's very well documented because it's giving rise to a disease. I don't remember the name, but I can find it back. Sorry, it's in French, but it's quite easy to understand. This is the nucleus, this is the mitochondria, this is before editing, and this is after editing. So here you have a tRNA. It's the tRNA that is normally used to transfer the tryptophan during translation, and the anticodon sequence is CAA. The number 34 is just the position. Okay, so CAA. Naturally, the nucleus produces this tRNA like that with CAA. So before edition, yes, we have to read it in the other way, okay? It's CCA. Before edition, the anticodon is CCA. You see the 34 is here, and the codon that is going to be read is GGU, okay, it's like that. But instead of producing another tRNA with a different anticodon, so in that case, it is UCA and not CCA, because the C can be converted to a U, okay, chemically. I mean, it's by an enzyme, but it's not during transcription. So it means that the nucleus produces one variant only of this tRNA tryptophan. But this tRNA tryptophan, when it enters mitochondria, because this is where the reaction happens, the tRNA, which is CCA, can be turned chemically to UCA. And then it means that this original tRNA that was normally, it carries a tryptophan. You understand that? But it will, during translation, add a tryptophan for a different codon. You understand? So this is one aspect of RNA edition. It means that, so you see the world of RNA is very complicated because after transcription, we talked about transcription, but after transcription, there are so many processes at the RNA level. You have the cap, you have the polyadenylation tail, you've got the splicing. In the splicing, you have alternative splicing. And after, even when you look at the sequence of an RNA, you have to keep in mind that the base on the sequence of this RNA can also be chemically modified by an enzyme, not by transcription, to have change of the base. So it means that the final RNA, they often don't have the same sequence as the originating RNA because of addition. So it makes a lot of possibility. And there is it's still in French, but don't worry. There is a very well documented case in human. It's about a protein. I'm not very familiar with the disease in human, but I know that this protein, when it's mutated, it gives rise to disease in human. Anyway, there is in the same idea as just what we saw for alternative splicing. Remember, what we saw for alternative splicing is with one gene in the genome, in two different organs, we had two different spliced versions of the transcript. We can go one step further. In that case, in that example, with one gene, which is the one on the top here, so all the purple, pink boxes and all the little bars are the exons. So they are very small exons, plenty, one big exon, and so on. So translation start, stop, the transcript, mRNA, the ATG, the stop codon. No, stop codon is here, and this is one codon inside this exon. So the genome contains this copy only. Once this gene is transcribed and spliced, there is only one messenger RNA that is produced. So there is no alternative splicing. One gene, transcription, capping, polyadenylation, splicing at the same time. You've got one mRNA. And this mRNA, so capping, épisage in French means splicing, and polyadenylation means polyadenylation. So all the process for a normal transcript, but depending on which organ it is, so intestine in French is intestine in English, foie in French is liver. Okay, so this messenger, if it's produced in GUST, in intestine, or in liver, the addition of the RNA will be different, and this addition is particularly drastic because in the liver, there is no addition. Pas d'addition, no addition. The sequence CAA present in the messenger is kept and the protein is translated as it is. Okay? It's called ApoB, and the molecular weight of this ApoB protein is 100 kilodaltons. So the name of this version is ApoB 100. Okay, so transcription, splicing, so on, translation, 100 kb protein. But in the intestine, there is RNA addition specific to this transcript and specific to that point. The modification that is happening in this addition is the conversion of a C to a U, the cytosine to uracil. Instead of having CAA, which is a proper codon, it turns to be UAA, and UAA is a stop codon. So in the intestine, the protein that is going to be translated from this edited version of the mRNA is going to be translated, yeah, sure, but there will be a premature translation stop, and the protein translated will be stopped before the normal stop, and the molecular weight of this protein will be only 48 kilodalton. Okay? So do you see the similarities between what we just saw before, alternative splicing, which concern which part of the HN RNA you remove during splicing? So from that, you can produce different protein through alternative splicing. In that case, it's a little bit different, but the consequence is the same. We have one gene at the beginning, we can produce two different proteins in two different organs. So exactly the same situation as alternative splicing. But the difference, it's not alternative splicing, it's RNA edition that introduced a stop, a premature stop, within the open reading frame, and so we can have two different proteins in two different organs. Okay, so you see the conceptual similarities between alternative splicing and RNA edition. At the end, we can have different proteins from a single gene, but the process to obtain the two different proteins is different. Is it clear for you? Yes or no? If you have any question, do not hesitate. Huh? It's okay. So we can go on. So a summary complete for all what is just a reminder is to keep in mind that this is RNA pol 2, which is transcribing the RNA, which is there. This is the C-terminal domain, and the RNA is here, okay, in red, 5 prime. Just to show you, and this is what I wrote as a summary for, sorry, I need to put back my mouse. And this is the summary of all what we saw, is that, okay, all the splicing, polydelineation, and I forgot to say capping binds to this C-terminal domain of the pol 2, and all those processes, capping, splicing, polydelineation, and editing, I should have put editing, start before the transcription has already finished. So all those processes that we see, they are happening at the same time during transcription elongation. Okay? So this is just a summary of that. Okay. Okay, now we're going to, so we have half an hour for this. We're going to see, so before that we start this, do you have some question about this general presentation of all the polymerase and the different RNA processing? Is it okay? Because now we are going to start a different part of the lecture is from all what we know about the theoretical mode of action of the pol 2, how now technically in the lab we can study the promoter and the transcription factor that are going to be responsible for transcriptional activity of pol 2. Do you have any question? Otherwise, we start with more technical things. Is it okay? Okay, so we can start. So now, and this is what we are going to go on with the exercise on Monday. We're going to study, so I will show you now one specific example that was obtained in plant. In Monday, we are going to work in growth hormone regulation, the gene which are the target of the growth hormone in mammals. So we are. So this is just a drawing to show you the diversity of eukaryotic pol 2 genes from mammals to the less evaluated one, so Saccharomyces. So just to show you the promoter, and after we finished all that, in mammals, the structure of the gene, they are much more complicated than in Saccharomyces. You've got all the details. That's for your own information. Some in Saccharomyces, you don't necessarily have the data box anyway. So we're going to study, I put this drawing here. So you know that all the RNA Pol2 complex binds this region around the transcription initiation start. And we are going to study all the upstream regulatory sequences which are responsible for the activity of the polymerase in specific situation. So there is inductibility, there is tissue specificity, and so on and so on. So we are going to see how we can study the cis-acting elements, so the bits of DNA that are responsible for activity of the promoter, and the trans-acting element, which are the transcription factors specific to one set of genes. So, the topology is like that. We are not going to study this part. This part is the pre-initiation complex, so all the factors that we saw before, Tata box, Pol2, and so on. This is the machinery that is going to be able to produce the RNA. We are going to focus on the upstream or downstream. It's important to have that in mind, because regulatory sequences and protein can be upstream. Most of the time it's upstream in the promoter sequence, but sometimes we can find them downstream, or sometimes we can find them very, very far away from the promoter. So there is something that is quite important. So the topology of a promoter is very, there is no common feature. We have always this core promoter element, which is the site of binding of RNA Pol2. Then in this drawing, you see that they put regulatory sequences upstream and downstream. And there was an extremely clever analysis. It's the distance of this regulatory sequence relative to Tata box. What is the importance of the distance? So before that we go on, I shift to the things to show you something different. Okay. So one feature, so I will draw you a promoter. So here we've got the transcription start, and let's say this is the first exon, and so on, we go on. Okay, and it's going on. So here we've got the Tata box, but we are not interested, and this is all the pre-initiation complex. You know that there is the C-terminal domain of the RNA Pol2, and this is the one that needs to be phosphorylated in order to activate transcription. Okay. This activation, this phosphorylation is mediated through proteins that can specifically bind cis regulatory element, and the protein, we call them trans-regulatory elements. How a protein that can recognize DNA can activate the RNA Pol2? It has to enter in interaction in order to trigger the phosphorylation. So what I want to talk to you about is the space, the distance between the core peak and the cis regulatory element, because it's quite complicated in the coding region. The coding region is everything after the transcription start, because this is the sequence that you are going to find back in the mRNA. In the coding sequence, So I take my mouse. It's quite. You see the mouse? Well, okay, I suppose yes. So in the coding region, every single nucleotide is important because the size is very important. For example, if you introduce one single mutation in an exon, you won't have the same protein. So the structure, the topology of the coding region is not flexible at all. Okay. But there is something completely different for the promoter. Everything that is upstream the transcription start, we have the data box here for the data for the polymerase to binding, but a cis regulatory element that is upstream. It can be 1 kb away from the data box because, so I just draw you another. So here we've got the pre-initiation complex, but even if the cis regulatory element is very far away, so this is the transcription factor, the DNA, this bit of DNA is completely flexible and can bend, and so the transcription factor can interact with the C-terminal domain and phosphorylate. Okay? You understand what I mean or not? Is it okay or not? So, what I want is quite difficult for me to explain. For what I want you to remember is everything before the +1, you can have regulatory sequences, but the place where they are relative to the RNA pole is not very important because DNA can bind and the interaction can still happen. Another thing, so keep in mind in promoter, the distances, they are not very crucial. Another point, it is the orientation of the sequences. So I show you one example. You have one promoter, so the coding region, the peak is here, and we've got one regulatory sequence in that direction. There are experiments that show that same concept, the peak is here. If a promoter contains the same sequence, the distance at the same distance, okay, but that the sequence orientation is reversed compared to the other, it generally it won't affect the binding of the protein. The binding of the protein will just be reversed, but the protein can still manage to phosphorylate polymerase and activity. So you can imagine something for the promoter, which is like that. And after we will see the rest of the slide. One gene, so I try to draw it in the scale. Okay, so this is in red, it will be the peak. Okay. The second artificial gene, exactly the same. So I try to be okay. The peak is exactly at the same place. Okay. And this is the +1. We can have, no, sorry, I'm going to draw you 3 situations. So exactly the same. Okay. So I'm going to show you 3 situations in which the gene can be active, so transcribed. You can have one cis element here in the first case, that is going to be recognized by a transcription factor, and because DNA can bend, activation is okay. All right? Do you follow me? Yes or no?
Yes.
Okay. You can play, and I will show you real experimental data after. You can play, so let's say the distance between peak and TF is X. You can create an artificial promoter where you put more space between peak and the transcription factor. So we have X that is bigger than X, okay? In that case, the gene can be also active because DNA can bend, and so there will still be activation, okay? It seems to be very simple, but it is the case. Third configuration, so sorry, I will put it here, we put it in that direction. And here we put it in that direction. We can have this situation, so the sequence, the cis regulatory sequence is exactly at the same place as the first case, but the orientation is in the other way around, okay? It's not a problem because the transcription factor can bind, and because it can be close to, it can still activate. So you can have this kind of situation. Okay, so if you look at carefully this awful drawing, sorry about that, the localization of the site of the binding of the transcription factor doesn't have a big impact in terms of activation because DNA can bind, okay, can bend, sorry. And second point, the orientation, sense or reverse of the sequence still allow the binding of the transcription factor and therefore can still activate the POL2. So you see, when you try to analyze the regulatory sequences inside the promoter, the topology is very, very plastic. The regulatory sequence, you can find them at completely different places. Okay? Do you understand this concept? So, as a general rule, the distance doesn't matter and the orientation doesn't matter. So that's theory. But in practical, there is something that has been modelized. No, sorry, it's not this one. Okay. So it's a little bit technical. It's very complicated, but I hope you can. So you can read carefully all the texts during this weekend. I will try to explain you what does it mean. Okay, all these graphs there, and we will talk about that later on Monday. If you didn't understand what's happening there, because you will see there is an example after that clearly demonstrates this modeling analysis. So I explain you a little bit the story. Quite difficult to understand the way they draw the way the drawing is done. So we are going to start with the right part of the graphic, which is the more understandable. What they represent by this green line, it's written effective concentration. So I will change completely this for you to understand. So you see in the drawing on the left, there are, this is the piece of DNA, okay, whatever piece of DNA, and the two little balls, the white and the red, there are two proteins that are bound to two different sites of DNA, okay. So so it's a pure mathematical modelization, this, but based on real experiment. So what they did, the graph on the y-axis, it is the probability that the white protein can get in contact with the red protein. In other words, the higher it is, the more chance you have to have the red that can get in contact to the white. Okay? And on the x-axis, it is the distance, the physical distance in, so it's written site separation in nucleotide pairs, is the base pair distance between the red and the white site. You understand the concept of the experiment. So y-axis, the probability of having red touching white, and in x-axis, the distance, physical distance of white compared to red. Do you understand the graph? Okay. So if you had to analyze this graph, it means that we are checking for the probability of contact according to the distance that separates the two objects, the red and the white. So we are going to talk about this little bit at the very beginning after, because it's very complicated and it's very interesting. So what you can read from this graph is that two sites where two proteins can interact that are separated by 500 base pair. So two sites in the DNA separated by 500 base pair. The bending capacity of DNA is such that if they are separated by 500 base pair, you've got almost 100% chance that because of the bending, they can touch each other. You understand? Or no? Okay. After, if we move a little bit along the x-axis, it means that we increase the distance between the two sides. And what you realize with this graph is the maximum of the probability of meeting of the two proteins is 500 base pair of separation. And if you increase little by little this distance, they can still interact because DNA can bend always. But because they are going to be far away little by little from themselves, the probability is a little bit dropping. So this is what we see here. The maximum of probability of meeting for two proteins is when they are separated, when they are on DNA, it's when they are separated from 500 base pair. Then the more you separate them, the less probability you have to have contact. Okay, so that makes sense. But what is really weird is this part. So this part is if the distance between the two proteins is very short, we can imagine that it would be easy for them to get in contact, but it's not the case. Why? Alors, this part, this dropping part of the curve, is because you have two proteins. Okay, what I'm going to do is I'm going to draw something. It will be easier to understand. So when you decrease the distance between two factors, so first situation, we've got one element here, and the other one is separated by 500. So this is the maximum. So it means that the DNA can bend very easily, and the two sides, they can be very, very close together. After, if we go on and we have them more close, so let's say 100 base pair of distance between the two. The DNA can bend, but not completely. So it means that because it cannot bend completely, the two sides, they cannot touch each other very frequently. Okay, so we can imagine that if they are closer, they can interact more easily, but actually it's not the case because they are close, but not close enough. And the capacity of bending of DNA is not enough. So it doesn't work. This is why we see the curve that is completely, so the curve is like that. And it's completely dropping. This part is because the bending is not necessary. And then there is this part of the curve that is completely flat. There is no curve anywhere. So I have to explain you how it works. In the DNA, okay, it's awful, my drawing. It's a double helix, okay, with a minor groove and a major groove, okay? Sorry, but I need to redo some kind of nicer. Okay, so this is the minor groove of DNA and this is the major groove. Okay, you know the double helix, there is one small groove and one big groove. What's happening is that and last point, one turn of the helix, okay, one turn is 10 base pair. Okay, so we have one turn every 10 base pair of DNA. So it means that the major grooves, this one and this one, are going to be on the same face of DNA every multiple of 10, every turn, okay? And what's happening is that most of the protein interact with DNA, interact with the major groove. Okay? Do you follow me? Because it's quite tricky, to be sincere. Is it okay? Yes or no? Or do I lost you completely?
Still following you.
Sorry?
Yeah, it's okay. You still understand all the time.
You understand? I'm trying to explain you what's happening for very short distances when this number is very, very short. Okay, so okay, what I'm going to do, I'm going to redraw it because, so I'm going to show you like this. Okay, it will be easier. Okay, so this is DNA and the interaction of two proteins, it's only in the major grooves. Okay, and we know that one turn of DNA represents 10 base pairs. Okay, so what does it mean? It means when two sites of protein DNA interaction are very, very close. We are talking about number less than 100. Okay, when two potential sites are very, very close, let's say 50 base pair, you've got one binding site for one transcription factor, and 50 base pair away from that, you've got another binding site. They are separated by 50 base. Okay, 50 base is a multiple of 10. Do you agree? So it means that if you check for 50 base, you've got 5 turns of DNA, and 50 base away, you have the two major grooves of DNA which are on the same side. Is it okay? Yes or no? Tell me, because if you don't understand, I will try to find other material to show you again.
So this is between two turns of the DNA?
This is one turn.
One turn.
Okay. Okay. Here we start, we've got half a turn here because the major groove here is at the bottom. Here it's on the top. And here it's at the bottom. So from this place here to the other place of DNA, we've got one full turn. 360 degree, okay? One turn. This is 180 degree in the middle. If you do one turn, if you check one turn of DNA, you've got the two major grooves, the only major groove is exposed on the same side of DNA. Okay, because we did one turn, one complete turn. Understand or not?
Yes.
Can I ask what is the major groove?
Ah, okay. You don't know the structure of DNA? There is two grooves. The double helix. You know it? Okay.
Yes,
One.
helix structure.
What?
I mean, I know the helix structure, but I'm not sure about the major groove.
But so you know, well, okay. 1 second. I open another. Okay, I just opened another PowerPoint file that I have. I think it's not in French. 1 sec. Okay, I've got it. 1 second. I share it and. I lost my Zoom.
I'm sorry, you are muted. I'm sorry, I cannot hear you. Maybe because your mic is off.
Is it okay now? Can you
Yes.
hear me? Okay. I am fed up with Zoom. Okay, it was this one. So I just share. So now, do you see the structure of the double helix? Yes or no? Can you see it or no? Okay, perfect.
Yes, you can see.
Okay, no, because if you don't tell me, because I've got technical problems. So the major groove, look at there. So you see the blue spiral and the red spiral. You know that the minor groove is this groove, which is really smaller than this one, which is bigger. You know that? Yes or no? What's happening when you have protein that interact with DNA, like all the transcription factor, they most of the time interact with the major groove. It is the blue spot that you see here, so the protein can enter there and recognize the sequence here. It's never in that part because it's a minor groove. So what you have, the structure of DNA is like you need to have one turn to have, for example, if we start here, the major groove is exposed on the left. And you need to have one full turn to have the major groove re-exposed to the left. If you do half a turn, the major groove is exposed to the right. Do you see it? Yes or no?
Yes.
Okay. So this is now I shift back to the, and this is a very, it's very complicated, but it's very important. So if we go back to what I was drawing, I hope it works. Okay. So this is here. It's a protein that interact with the major groove, and if we do one turn, another protein that can interact with the major groove. So at that point, the two proteins, the two sites of interaction, they are separated by 10 base pair. Okay? If two sites are separated by 10 base pair, it means that the two proteins that can bind to these two sites, they are on the same side of the DNA. Do you agree? Because one turn brings you on the same side. Okay? So the drawing is not at all as the scale because the protein, they will be much more bigger. If they are on the same side, what does it mean? They can interact. Okay? Each other. Do you understand? Okay. If the two proteins, the two sites of protein are separated by 5 base pair, it means one protein here in the major groove. So from here to here, it's 10, but from here to here in the middle, it's 5 base pair. So if two proteins, two sites of interaction are separated by 5 and not 10, it means that the two proteins, they are in interaction, but in the two different sides of DNA. Do you understand? It's on the other way.
Yes.
And the consequence is that because they are not in the same side, they cannot enter each other. You understand? So the consequence of all that is this. If you have, so I will explain you what so I redraw the graph and I will write 0, 5, 10, 15, 20. And here it is always the probability of contact between the two proteins. If two proteins are separated, two sites of interaction of proteins are separated by 5 nucleotides. The two sites are on two different sides of DNA, so no interaction. Okay, so I write it like that: no interaction. If the two sides now they are separated by 10 base pair, it means that the two sides they are on the same side of DNA, which allows the interaction. So 10 base pair of spacing. Total probability of interaction. Then you separate by 15 base pair. 15 base pair is 1 turn and a half. So it means that if two sides are separated by 15, the both sides are on different sides. So 0 interaction. Then if you have the two sides separated by 20 nucleotides, 20 nucleotides means 2 terms. So 2 terms means the two major grooves on the same side. so the protein can interact because they are on the same side, and so on. So maximum, and at 25 it will be minimum, at 30 it will be maximum, and little by little after we start with the normal curve. Do you understand this? No interaction, interaction, no interaction, interaction, and so on and so on, because no interaction is when the proteins are on opposite side, and interaction, they are on the same side. And shift like that every after. So to come back to the graphic, maybe it will be more clear. Anyway, I hope it's this one. Okay. So the graphic was like that. So we are talking about only this graphic. So look, this is what 500. I told you if the two sites are separated by 500 base pair, there is total probability that the two proteins can get in contact. After, if you extend the distance between the two sites, the probability is reducing slowly by slowly. After, from 500 to 100 base pair, it was dramatically dropping because of the constraint of bending of DNA, because DNA is flexible, but not that much. So at one point, it cannot bind, it cannot bend. So here it was impossible. But you see, what I wrote on the whiteboard, zero maximum every term. Here, I don't know if you noticed, but you don't have any green line here. Because so if you check really well, every unit is 50 base. From 0 base to 50 base of spacing between the two elements, the two sites of interaction, it is 100% if it's one turn, and it is 0% if it is 1 turn and a half. So the beginning of this probability curve is maximum, minimum, maximum, minimum, maximum, minimum. Then DNA can bend a little bit, then it can bend very easily to arrive to the maximum, and then it drops down a little bit because the distance that is going very, very high, it's 2 kb there, so it's very far away, reduce the probability of meeting. Do you understand that? I know it's quite complicated because it's a conceptual modelization of the probability of interaction of two proteins bound to DNA according to the distance of the site of binding. And there is quite a few. Well, anyway, we will see. Try to think a little bit about this during this weekend. We need to finish now. We are going to use this concept. So what I would like you to do, I explain you, we take 5 minutes on what we are going to do for next week. You try to understand, and if you have all the questions, you can write them down, and we'll talk about that on Monday, to understand this graph, and especially what's happening here in this region where there is this same side, not the same side, not the same every term. This is going to be important to understand the following. So you try to think a little bit. If you don't manage to understand, don't worry. We will take it. I will explain it to you again. And then we are going to, and please take a look at the following slides. I just tell you, so this is part of the work, not this one. This one, where we're going to analyze the structure of a promoter, really in depth. So this is a published paper. And if you look at, so I just want that you pay attention this weekend to this graph, okay? and especially that part of the graph that I try to explain you, but I will take it again on Monday, and try to put it in relation with the analysis of this construct. Okay, and you will see that concerning the result, this result especially, in this strategy, we are going to identify precisely the cis regulatory element of a POL2 promoter that is able to direct tissue-specific expression. The example is from a plant, but it's the same for animals. So we're going to see all the technical strategy to analyze the sequence, but in order to be able to have a fine analysis, you need to understand a little bit this part of the curve, okay? So it's not too much work. You try to think about this slide, try to understand. If you don't understand everything, don't worry, we will take it completely from very in-depth, and try to find the relation between the results on this slide with the conceptual modelization that is presented only in the graph, and don't look at the blue circles and so on. Okay? Is it okay for you? Okay?
Yes.
Yes.
Okay. So, we will do that. I know, I understand that you don't really understand what I want to present you. Don't worry, Monday we will sort everything, because you will understand that this precise part of the curve means something very important in terms of polymerase II gene regulation. Okay, and I will explain you how it works. And after, you will see, when you will have understood this slide, you will have understood everything. And then we will do the exercise on the mice and the growth hormone. Okay, so we will do that on Monday. Okay, so we have finished for today. And same situation, if you have any question, we can take time on Monday to answer that. And on Monday, we will start at 3 p.m. for you, so 9 for me, 9 a.m. for me. Okay? Perfect. So I wish you a good weekend. And we meet back on Monday morning, Monday afternoon for you, and Monday morning for me. Okay, I have to leave you because I've got another lecture in half an hour, not quite far from here, actually.
For the Monday exercise, so what we will divide in group, or we will work.
No. On Monday, the exercise. So, Miss, do you use Moodle, the Moodle for sharing data? Moodle at USTH?
Not very often.
Not very often, but don't worry. So, what we are going to do on Monday, Hang will send you the exercise, and we will do step by step. It's okay. It's under the form of an exercise, but it's a complete study of one promoter. It's a very interesting, you will see it's very, very interesting. And what we are going to do is, all together, we are going to answer question by question and try to understand how this promoter is working. And you will see it's quite complicated. But we will do all the class all together, and question by question. Okay?
Yes, it's okay. Thank you.
So the exercise, you will have it on Monday morning for you, but don't worry, you don't need to prepare it, because we're going to do it all together. Okay? But nevertheless, for Monday, try to look at the 2 slides that I showed you, and try to, if you don't manage, don't worry, I will explain you. But if you find the relation, it will be nice. Okay?
Yes, okay.
So have a nice weekend and see you on Monday, 3 p.m. for you and 9 a.m. for me. Okay. So I have to leave you quickly. Sorry about that.
Okay, see you on Monday.
Bye-bye. Bye-bye.
Thank you.